Hi, Bio 495 and 595. My name is Dominic Colangelo,
I'm Jessica
and
Wilson.
today we're going to go over changes in tropical leafing behavior with climate change over 9 decades, a case study from the Singapore Botanic Gardens. We'll be discussing a fascinating study on plant phenology. Plant phenology is the timing of key life events such as leaf flush, leaf fall, flowering, and fruiting. It's a fundamental component of how plants interact with their environment. And these events regulate not only individual plant fitness, but also shape plant-animal interactions, species distributions, and ecosystem processes. While phenological research has historically centered on flowering and fruiting in crops, global climate change has elevated the importance of understanding phenological shifts more broadly. Mounting evidence shows that rising temperatures are altering the timing of these events, especially in temperate regions, with cascading effects on biodiversity, nutrient cycling, and climate feedback systems. Tropical ecosystems, despite their rich biodiversity, have been underrepresented in long-term phenological studies. This is due to several challenges: greater species diversity complicating monitoring, rainfall rather than temperature being the main environmental driver of seasons, and a lack of long-term ecological research infrastructure. As a result, applying temperate-based phenological models to tropical systems can lead to inaccurate predictions. Yet understanding tropical phenology is increasingly urgent as these ecosystems face rapid climate shifts. As you can see from the images, tropical ecosystems are far more diverse, complicated in nature, and contain significantly larger amounts of above-ground biomass. Historical datasets offer rare insights. One such example is the work of Richard Holtham at the Singapore Botanic Gardens from the 1920s to 1940s. His systematic observations of leaf flush in cultivated tropical trees form a unique baseline against which present-day data can be compared. Leaf flush refers to the production of new leaves by a plant, often occurring in distinct seasonal cycles. It marks a key phase in a plant's phenology, reflecting responses to environmental cues such as rainfall, temperature, and day length. With the recent revival of phenology monitoring in Singapore, we are now able to ask critical questions. Let me turn it over to Jessica now.
These are the three research questions from the study. Have leaf flush intervals changed between Holtham's time and present day? How do rainfall patterns affect leaf flush? And have rainfall patterns changed between Holtham's time and present day?
All right, now let's move on to the materials and methods. This study was conducted in the Singapore Botanic Gardens, located just north of the equator. The gardens experience a tropical rainforest climate with consistent day lengths and warm temperatures year-round. This is punctuated by two distinct monsoonal periods: the wetter northeast monsoon from December to March, and the drier southwest monsoon from June to September. These seasonal shifts, especially in rainfall, are key environmental cues for plant phenology in the tropics. The gardens themselves, founded in 1859, are an ideal site for long-term phenological research with rich historical records and a stable landscape. Holtham conducted his leaf flush observations between 1927 and 1940, recording the dates when trees produced new leaves after a full leaf drop. The study used his original rainfall and phenology data, updating species names to align with current taxonomy. In the present study, which began in 2016, 237 specimens were monitored from 90 species, aiming to match Holtham's original trees or include the same species. Weekly observations tracked 6 phenophases, though only leaf flush was analyzed here. To compare historical and modern data, generalized linear mixed models to test if leaf flush intervals differed between the two periods. In addition, sliding window analysis was incorporated to assess how rainfall affected leaf flush timing, searching for the most predictive time window prior to each observation. Lastly, the authors evaluated changes in rainfall patterns themselves using generalized additive models. These were compared using model selection to assess long-term shifts in tropical phenology and link them with environmental drivers, especially rainfall.
The results from the study are organized by research questions. The first question is, have leaf flush intervals changed between Holtom's time and present day? Leaf flush intervals between the two time periods have shortened considerably, from 9.6 months to 6.4 months for the species that are in both datasets. Out of all the species studied, leaf flush intervals increased for only 3 species. This figure shows the difference in leaf flush intervals for all the species in both studies. The red represents Holtom's time and blue represents the present day. As you can see, the intervals from Holtom's time were longer on average than present day. There are two explanations for differences in leaf flushing intervals from Holtom's time to present day. First, leafing is associated with rainfall, and second, there are 2 drier and wetter periods a year in the present day, compared to 1 drier and wetter period a year in Holtom's time. However, in Singapore and other similar climates, there are not actual wet and dry seasons, but rather wetter periods and drier periods. The second question analyzes how rainfall patterns affect leaf flush. These results are for species in both datasets. For 5 of the species, leaf flushing was associated with less rain. This can be found at the bottom of the figure where the leaf flush coefficient is less than 0 for all data points. For 4 of the species, leaf flushing was associated with more rain, as seen at the top of the figure. For the remaining 14 species, there's no strong association either way, because the leaf flush coefficient overlaps with 0. An explanation for why the results for how rainfall affects leaf flush are so varied is their complex tropical phenology. Tropics are very biodiverse and complex. For the 5 species, leafing was preceded by periods of drier weather. This is because dry weather triggers leaf fall, and plants produce new leaves following leaf fall. This is also a defensive adaptation. Insect herbivores are suppressed during dry periods, so plants produce new leaves then to avoid predators. For the 4 species, leaf flushing is preceded by wetter weather. A possible explanation for this is that plants require water for new leaves, so an increase in water leads to an increase in leaves. Question 3 asks, have rainfall patterns changed between Holtom's time and present day? As previously discussed, during Holtom's time, there was 1 wet period and 1 dry period in a year. Now, there are 2 wet periods and 2 dry periods. This figure shows the distribution of rainfall throughout the year, with Holtom's time in red and present day in blue. In the present day, the dry periods are February and August, and the wet periods are May to June and October to December. In addition, in the present day, the dry period is drier and the wet period is wetter. The dry period in February, as shown in this figure, has decreased in average rainfall over the last 100 years, and the opposite has happened for the wet period in May. Seasonality is increasing in other parts of the world, including the Amazon rainforest in Brazil. As a result of increasing seasonality, leafing cycles shorten, and leaves fall and regrow more rapidly.
All right, so in conclusion, our study shows that tropical trees at the Singapore Botanic Gardens are leafing more frequently today than they did nearly a century ago. And changing rainfall patterns are likely a key reason why. This shift in weather patterns appears to be accelerating leaf cycles in many species. We also found that some species responded to wetter conditions before flushing leaves, while others did so after dry periods. This variation highlights the complex and species-specific nature of tropical phenology and the need for more tailored modeling approaches in future work. Beyond rainfall, factors like surface light intensity and cloud cover may also be influencing these patterns, though they weren't available in our data sets. Importantly, faster leaf turnover could have wide-reaching implications for forest health, nutrient cycling, herbivory, and even carbon sequestration. Yet much remains unknown, especially in the tropics where long-term data is scarce. By combining historical records with modern monitoring, this study demonstrates how botanical gardens can serve as powerful tools for climate change research. Looking forward, establishing coordinated networks of tropical gardens could expand our understanding of how global change is reshaping tropical ecosystems. The phenological patterns observed in cultivated specimens within botanic gardens may differ from those seen in natural forests, where trees grow in denser, more competitive environments. However, the accessibility and controlled setting of botanical gardens offers significant advantages for long-term monitoring. With fewer logistical challenges and lower resource demands, researchers can observe a wider range of individual and species. These settings also lend themselves well to citizen science initiatives and data collection through public contributions. Thanks to the high number of visitors and Garden's strong emphasis on education and public engagement in botany and environmental awareness. Establishing a coordinated network among tropical botanical gardens, similar to existing collaborations in other regions, would enable the scaling up of phenological research. Such networks could support broader investigations into regional trends and facilitate hypothesis testing across diverse ecological and climatic contexts. So while this isn't just academic, changes in the timing of leaf flush can ripple throughout ecosystems. This affects food availability for animals, plant reproduction, and forest health. In urban tropical settings, climate change is layered with urban heat island effects, making botanic gardens powerful sentinel sites to detect these changes.
This study is significant for several reasons. Plant phenology monitoring is rare in the tropics, mainly because they were initially considered aseasonal and sometimes still are. When we think of changing rainfall patterns, we don't often think of the unchanging tropical rainforest. This study is unique because it spans 9 decades. This was made possible because the Singapore Botanic Gardens have been well documented since the 19th century. Predictions for plants in tropical regions that are based on studies done in temperate regions can be incorrect, so it's important to study the tropics themselves. Botanical gardens have diverse taxonomic and geographical groups, which are valuable in studies like these. There are major biological implications for the results of this study. Higher seasonality, as mentioned previously, results in shorter leafing cycles and more rapid leaf turnover. This leads to changes in trophic interactions and nutrient cycling, which affects plant health and ecosystem resilience. The rate of decomposition can also become slower than the rate of nutrient depletion. When leaf flushing occurs, plants require more nutrients to build new tissue. The decomposition of fallen leaves may take longer than nutrient uptake, and this leads to temporary nutrient stress. Tropics are essential for Earth's biodiversity, so these outcomes affect the whole globe. While this study is significant, there is still more research to be done. In this study, all the species responses are grouped into one, and it would be useful to have models that allow species to respond differently to weather changes. Another study could examine if plant traits explain species-specific phenological responses to weather. Traits that reflect mechanisms of adaptations to drought, such as embolism resistance, rooting depth, and water storage, would be beneficial. Other weather variables, such as surface irradiance, could be studied as well. For example, the intensity of light that reaches tree canopies, which indicates the amount of light available for photosynthesis. Lastly, consistent monitoring over the span of the study instead of having two studies at the beginning and end would be helpful. Thank you for your attention.